in dicot stem. So, in the slide, the first image is shown the primary structure of dicot stem. Already we have discussed in previous sessions. The dicot stem shows, the vertical section of dicot stem shows three parts: epidermis, cortex, and stele. Stele occupies large space at the center. It's like a central column. It's having a limited number of vascular bundles. So these are vascular bundles. The number of vascular bundles are limited, arranged in a circle. Such type of stele is called U stele. U means true stele. And each vascular bundle is conjoint, collateral, and open. That means in xylem and phloem are separated by a strip of cambium. This cambium is called vascular cambium. This in each vascular bundle you can see a green strip. That green strip is nothing but it is a vascular cambium. This vascular cambium is nothing but it is a meristematic tissue. It helps in secondary growth. So let us go in detail. So this is the primary structure. Already we have discussed epidermis, cortex, stele. And each vascular bundle you can see collateral, conjoint, collateral, and open type of vascular bundles are there. Here, see. Here in this image you can see a blue color strip. Generally, in higher plants, particularly in secondary growth is seen only in dicot plants. Actually, it is absent in monocots. In flowering plants like dicots and gymnosperms, you can see the secondary growth. Since the vascular bundles are open type of vascular bundles are seen in both dicotyledons and gymnosperms. These both are flowering plants. But in monocots, the secondary growth is absent because the vascular bundles are closed type. There is no a strip of cambium is seen. 1 minute. Anna. Hey, mummy gave beta. Mummy gave call. Mummy gave. Metro बजना को call. Right. हाँ. मम्मी गई. हाँ. मम्मी गई. मम्मी गई ना. हाँ. Right. Okay. Sorry for the disturbance. It's an urgent call. So this is a primary stem. A strip of cambium is there. In monocots, it is absent. So let me clarify what is secondary growth. Generally, in plants, in flowering plants, there are two types of growths will be there. Two types of growths. The first type of growth at an early stage, it is seen. That growth is called primary growth. The vertical growth of the plant body is called primary growth. Primary growth is due to apical meristems. So in meristematic tissues, there are three kinds of meristematic tissues are there. Apical meristem, lateral meristem, and intercalary meristem. The primary growth is because of the activity in apical meristem. Apical meristem is present in the tips of roots and stem, shoot system. All the tips of shoot system and root system, you can find apical meristem. Because of the activity of apical meristem, plants grow vertically. That vertical growth of the plant body is called primary growth. Primary growth. The secondary growth, due to primary growth, the parts of the plant body like branches, leaves are increased. The number of plant pods are increased. The number of leaves and branches, they start producing flowers. That's why these are the parts which are increased due to primary growth. If the number of parts are increased in the plant body, the requirements are also increased. It needs increased number of pots, need more supply of water and more supply of food materials. Hence, the secondary vascular tissues are formed. The secondary vascular tissues are formed at later stage because of the activity of lateral meristem. The secondary growth is initiated. Because of the secondary growth, new or secondary xylem and secondary phloem are synthesized or formed. Because of that, the girthness of the plant body, the thickness of the plant body, the radial growth is seen. So that growth is called secondary growth. That means secondary growth is because of the activity of lateral meristem. The growth is seen after the primary growth. At the initial stage, in the initial years of the plant life, generally in almost all the plants grow vertically. That is primary growth. At later stages, the secondary growth is seen because of the activity of lateral meristems. Okay, see. So this is secondary growth is initiated here. Increase in width or increase in girthness of the plant body is called secondary growth. Secondary growth is carried out by the two cambiums. That means two cambiums are initiated. The formation of two cambiums are seen. The first cambium is vascular cambium. This is a blue inside the steel. Vascular cambium is formed. Vascular cambium is formed because of the activity of vascular cambium. The increase of the xylem and phloem is seen. And the next cambium, the outer cambium in the epidermal region, the outer cortical region, the one more growth is seen. That is because of the carp cambium. I'll repeat once again. The secondary growth is initiated by two meristematic tissues. The first meristematic tissue is vascular cambium and the second one is cork cambium. Vascular cambium is present inside the steel, and the cork cambium is outside, just below the epidermis, that is outer cortex. Let us see the next slide. Normally, secondary growth takes place in roots and stems of dicots and gymnosperms. Already we have discussed at this point. In roots also like stem, in roots also secondary growth is seen. But in monocots, secondary growth is absent. Why? Because there is no cambium. Since vascular bundles are closed, there is no strip of cambium present between xylem and phloem. That's why the secondary growth in monocots, almost all the monocots is absent. But exceptionally, some of the monocots like palms, yucca, dracaena, agave, and coconut. That's why the trunks of these plants are woody. Woody trunks are seen. In woody monocots, you can see the secondary growth. These are the exceptional cases. That means exceptionally in some, very rare, in some monocots, like palms. Palms means coconut is a well-known palm. In the other palms also, Victoria regia, that is an ornamental palm. Generally, it is seen in almost all the botanical gardens. Royal palm, sorry, not Victoria regia, royal palm. Royal palms are the ornamental trees. In those plants also you can find the secondary growth. In Yucca and Dracena, they are also present in gardens. Agave is a xerophyte. Almost most of the monocots show xerophytic characters. Agave is having a succulent leaves. Along the margin and tip of the leaf is having spines. So in these plants, you can see the secondary growth. Normally, in all the monocots do not show the secondary growth. Only palms like coconut, yucca, dracena, and agave show secondary growth. But almost in all dicots and gymnosperms, secondary growth is common. See, secondary growth is categorized into two types. There are two types of secondary growths are there. The first secondary growth is called intrastellar secondary growth. Intrastellar, the name itself is indicating. And the second growth of secondary stem is extrastellar secondary growth. I'll repeat once again. Intra, within the stele, there is a secondary growth in the initial stage of the secondary growth. The first growth is seen in the stele, inside the stele, because of the vascular cambium. Vascular cambium is present inside the stele. Because of the activity of vascular cambium, intrastellar secondary growth takes place. Intrastellar secondary growth is occurring. Because of this intrastellar secondary growth, secondary xylem and secondary phloem are formed. Along with the secondary medullary rays are also formed. Because of the formation of secondary vascular tissues, the primary vascular tissues and primary medullary rays will be crushed out, or they are replaced with the secondary tissues. This is called intrastellar secondary growth. Within the stele, the secondary growth will be initiated by vascular cambium. Intrastellar secondary growth is initiated by vascular cambium within the stele. Within the stele. In the later stages, extracellular secondary growth is seen. Extra means outside the stele. Outside the stele, that means because of the intracellular secondary growth, the central column, that is the size of the stele, increases. Because of the formation of secondary xylem and secondary phloem and secondary medullary rays, these are the secondary tissues. The primary tissues are crushed out or replaced with the secondary tissues. Because of the formation of secondary vascular tissues and secondary medullary rays, this stele size increases. Stele size increases. The central column increases, which because of it, because of the increase the girthness in stele, the epidermis, primary stem is protected by epidermis. That epidermis is ruptured. Epidermis is ruptured. It has to be replaced with secondary protecting layer called bark. That secondary protecting layers are formed due to extracellular secondary growth. It is initiated in the outer cortex. It is initiated in the outer cortex because the epidermis is ruptured when the size of the stele increases due to intracellular secondary growth. When the protecting layer like epidermis is ruptured, the secondary protecting layers are to be formed by the extracellular secondary growth. This extracellular secondary growth is initiated by cock cambium. Extracellular secondary growth is initiated by cock cambium. Cock cambium is formed. Some of the cells of outer cortex regain the capacity of division and become meristematic. They regain the capacity of division. That means they become again meristematic. Such meristems are called secondary meristems. The meristems which are formed at the later stages from the permanent tissues like parenchyma, collenchyma, such meristematic tissue is called secondary meristem. The secondary meristem, like cock cambium, is formed in the cortex region, particularly in the outer cortex. Some of the outer cortical cells become meristematic and forms cock cambium. That cock cambium initiates extrastellar secondary growth. Okay. How many types of secondary growths are there? The first one is intrastellar secondary growth. The next one is extrastellar secondary growth. Intrastellar secondary growth is initiated by vascular cambium. Vascular cambium is formed by the cambium, which is present between xylem and phloem, that is called vascular cambium. And primary medullary ray cells are also become meristematic. As a result, a ring of cambium is formed inside the stele. That is called vascular cambium. Because of the vascular cambium, secondary vascular tissues and secondary medullary rays are formed, and the girthness of the stele increases. When the size of the stele increases, when the radius of the stele increases, because of the pressure which is exerted by the stele, increased stele, the epidermis, which acts like a protecting layer in primary stems, will be ruptured. Then at later stages, this ruptured epidermis is replaced by secondary protecting layers. Secondary protecting layers. The secondary protecting layers are formed in extrastellar secondary growth. These extrastellar secondary growth is initiated by cock cambium. Extrastellar secondary growth is initiated by cock cambium. Cock cambium is a secondary meristem. Is a secondary meristem which is formed from the outer cortex. Because since the epidermis is ruptured, the outer cortical cells are exposed. Those cells which are exposed because of the rupturing of epidermis, those cells become meristematic. They regain the capacity of cell division and they become meristematic. The meristematic tissue which is formed in outer cortex is called cock cambium. Because of the activity of cock cambium, the secondary protecting layers are formed in secondary stems. So that is called extrastellar secondary growth. Let us see in detail. So this is primary stem. This is vascular cambium. A ring of cambium strip is seen. This is formed due to the fission of fascicular cambium and interfascicular cambium. There are two terms are there. Don't get confused. Already there is a strip in primary stem. Strip of cambium is present in primary stem. That strip is called intra. Within the vascular bundle, a strip of cambium is present. That is called intrafascicular cambium. This cambium. At the time of secondary growth, the primary medullary ray. This is primary medullary ray. The parenchymatous tissue. This is the primary medullary ray is nothing but it is an extension of medulla. This is pith. This is pith or medulla. The medulla is extended up to pericycle. between the vascular bundles. This is simple parenchymatous tissue. The cells of primary medullary ray, which are opposite to the fascicular cambium. This is fascicular cambium. This is fascicular cambium. This strip, the blue color strip, which is present between vascular bundles, become meristematic. This one. That is called interfascicular cambium. This strip is called intrafascicular cambium. This strip is called interfascicular cambium. Both the strips join together forming a ring. This ring is called cambial ring or cambium ring. This cambial ring is formed due to the fusion of fascicular cambium and interfascicular cambium. Fascicular cambium is also called as intrafascicular cambium. This fascicular and interfascicular cambial strips are fused with each other. They combine with each other, forming a ring. You can see the image in the slide. You can see the second image, right side top corner. So that is a ring of blue color ring-like structure is seen. That is called cambial ring. Cambial ring is formed due to the union of fascicular and interfascicular cambium strips. Fascicular and interfascicular cambial strips. Okay. The growth in this region starts earlier, where intrastellar secondary growth starts in the initial stage. A cambium that lies inside the vascular bundle is called intrafascicular cambium. This is a primary meristem, as already we have discussed. A strip of cambium already is existing in dicot vascular bundles. That's why they are open type of vascular bundles. In open type of vascular bundles, there is a strip of cambium present between xylem and phloem. That meristematic tissue is called primary meristem. Since it has not changed, it is not changed from the initial stage, from the embryonic stage. It has developed from the embryonic stage. The meristematic tissue, it remains as meristematic tissue even in the primary body. That's why that meristematic tissue is called primary meristem. That primary meristematic strip is called vascular cambium, which is present between xylem and phloem, is called primary meristem. And it is called as intrafascicular cambium or fascicular cambium. The medullary ray, which is extended from medulla to pericycle, is generally made up of parenchymatous tissue. The parenchymatous cells regain the capacity of division, which are adjacent to the fascicular cambium, forming interfascicular cambium strip. The fascicular and interfascicular cambium strips are joined together to form a ring of cambium. That ring of cambium is called ring of vascular cambium. This ring of vascular cambium produces secondary xylem towards the center, towards the inner side of the ring, and towards the outer side of the ring, secondary phloem tissue is formed. I'll repeat once again. Let me see the image. So this is the image. So this is outside the red color portion. There is a line is there, the black color line. That is vascular cambial ring, a ring of vascular cambium. This ring of vascular cambium produces cells towards inner side, inside the ring, and outside the ring. The more number of daughter cells are formed towards the inner side. They become secondary xylem tissue. Less number of cells are formed towards the outside. This light blue color portion is there now. So that is outside the cambial ring. That the tissue daughter cells which are formed outside the cambial ring are forms secondary phloem. That means The daughter cells which are formed towards the inner side of the cambial ring are secondary, they become secondary xylem elements. The daughter cells are which are formed outside the cambial ring are secondary phloem elements. That means secondary phloem is forming outside the cambial ring. Secondary xylem is forming inside the cambial ring. In vascular cambial ring, there are two different types of initial cells will be there. One kind of initial cell is called fusiform initial. The daughter cells are which are formed from the fusiform initials develop into secondary xylem towards inner side and secondary phloem towards outside. And the few cells in cambial ring, that is a ring of cambium, is seen. Some of the cells are ray initials since they form secondary medullary ring. Some of the daughter cells which are formed from the cambial ring from the ray initials remain parenchymatous. They do not develop into secondary xylem inside and secondary phloem outside. These lines are there, no? These lines are called these lines. These lines are called secondary medullary rays. The secondary medullary rays develop from the ray initials. This is secondary xylem. Outside, secondary phloem. Secondary xylem and secondary phloem develops from fusiform initials. Fusiform initials. Okay. Due to the activity of fusiform initials, secondary xylem formed inside, secondary phloem formed outside. Due to the activity of ray initials, xylem rays are formed inside. They are secondary xylem rays. And the phloem rays are formed outside the cambial ring. They are secondary phloem rays or secondary medullary rays. And inside, more number of daughter cells are formed. That means more secondary xylem is formed inside comparatively. The outside the cambial ring, less phloem, secondary phloem is formed. The amount of phloem is formed during the secondary growth is very less. And the quantity of secondary xylem, which is forming inside the cambial ring, is more. That's why secondary xylem is more, secondary phloem is very less. And this secondary formation of secondary tissues, that means the activity of the vascular cambium throughout the year is not same. As you know, during spring, new leaves and new buds and blooming is seen in plants, in flowering plants particularly. During autumn, they shed leaves, they shed flowers. When there is a formation of new leaves and new flowers, particularly during spring season, you can see the activity of cambium is more. Activity of cambium is more. They are very active. The cambial cells are very active during spring season because they need more supply of water and food materials since the new leaves are forming. But at the autumn season, in the autumn season, the plants lose leaves, they shed leaves, they shed flowers. They don't need much water and more supply of food is not necessary because the less number of plant pots are there. That's why the activities depending upon the seasons. During spring season, the vascular cambium is very active and produce more xylem and more phloem, more secondary xylem and more secondary phloem are formed because they need more supply of water and they need more supply of food materials during spring season. Since the formation of birds, the formation of new leaves are seen during spring season. That's why they need more supply of water and more supply of food materials. That's why the more amount of xylem and phloem elements are formed. That wood which is formed during spring season is called spring wood. Or since it is the early season, then that's why it is called early wood. It is also called as early wood. Since it is forming during spring season, then the wood which is called spring wood. The secondary xylem is generally termed as wood. Secondary xylem is generally known as wood. That's why the secondary xylem which is formed during spring season is called secondary spring wood or early wood. Let us check. And during autumn, already we have discussed the wood which is formed during autumn season. During autumn season is very less with narrow xylem vessels. Less amount of xylem and less amount of phloem is formed. Secondary phloem, secondary xylem are very less because plants shed leaves. Plants do not require the more supply of water and food materials. That's why less amount of secondary xylem and secondary phloem are formed with narrow lumen of the xylem vessels. That wood, which is formed at the later stages, is called autumn wood. Autumn wood or late wood. This is also called as late wood. Spring wood is called early wood. Autumn wood is called late wood. Late wood. Because in the later season, this wood is formed. That's why it is called late wood. Don't get confused. Wood is nothing but secondary xylem. Generally, secondary xylem is termed as wood. In the early seasons, that is spring wood, is large with wider lumens since they have to supply more amount of water and food materials. That wood is called spring wood and early wood. And later, in later seasons, plants shed leaves, particularly in autumn season. The wood is very less. That is called autumn wood. This autumn wood is also called as late wood. Early wood, late wood, or spring wood, autumn wood. These both woods represented, they represent the wood which is formed in a year. That means in 1 year, here you can see, in the image you can see, ring-like structures inside the steel. One is light in color, the other one is dark in color. Light color patches, rings are there, and dark color rings are there. This light color wood is spring wood, dark color wood is autumn wood. They represent the wood which is formed in a year. And together they constitute annual rings. Annual means in a year, 1 year. That means in 1 year, two types of woods are formed in the secondary stems. At the initial stage, spring wood is formed, which is light in color. Externally, you can see the light color. When you cut the logs, you can see a ring like dark rings and light color rings are alternate with each other. If you observe the logs, when they are cut, you can see the rings with different shades, light color shade and dark color shade. That secondary xylem, which is formed during early season, that is spring season, is called spring wood. It has wider xylem vessels a xylem vessels with wider lumen and light in color. That wood is called early wood. At later stages, at later seasons, autumn wood is formed. During autumn season, plants shed leaves. Plants do not require the supply of more water. That's why less amount of xylem is formed in secondary growth. That means it is inactive. Vascular cambium is inactive during autumn. That's why the wood which is formed during later seasons is autumn wood or late wood. How can you identify spring wood and autumn wood? In a log, you can see a different colored or shaded rings. Light color shaded rings are spring wood and dark color shaded rings are autumn wood. Spring wood and autumn wood constitute annual ring. That is representing the wood which is formed in a year. By counting the number of annual rings, you can estimate the age of the trees. After cutting them, by counting the rings, you can estimate the age of the trees. So there is a branch of botany which deals with the age of the trees. Age of the trees by counting the annual rings. The annual ring is nothing but annual ring which is formed, the wood which is formed in a year is called annual ring. That's why this, by counting these rings, spring wood, autumn wood constitute a wood which is formed in a year. That is generally known as growth ring or annual ring. Growth ring or annual ring. By counting these rings, we can estimate the age of the tree. Estimate the how many years old this tree can be estimated by counting those rings. This branch which deals with the age of the trees is called dendrochronology. Dendrochronology is a branch of botany which deals with the age of the trees. Age of the trees by counting the annual rings. By counting the annual rings or growth rings. Growth rings. So this is about annual rings. So see, autumn wood is a late wood. During winter or during autumn, very less amount of wood is formed, that is dark in color. See here, this is the section of the log, transverse section of the log. You can see here. This transverse section of the log, let me reduce it. See here. So these rings, by counting that each ring together, this is spring wood, this is autumn wood, spring wood, autumn wood, spring wood, autumn wood. They all alternate with each other. By counting these rings, we can estimate the age of the trees. That branch which deals with the age of the trees by counting the annual rings is called dendrochronology. So because of the secondary growth in stellar region, that is intrastellar secondary growth, the size of the stele increases. The central column increases in size. It occupies major part of the stem. Major part of the stem. Hence, because of the pressure which is exerted by the increased stele on the epidermis is ruptured. Epidermis is ruptured. And in place of epidermis, extra protecting layers are to be formed in place of epidermis. And new protecting layers are formed. So because of the loss of epidermis, the secondary stems has to replace this epidermis with the new protecting layers. By the growth is called extrastellar secondary growth. This extrastellar secondary growth is carried out by cock cambium. Cock cambium. This is cock cambium. Cock cambium is formed from outer cortex. Cock cambium is formed from outer cortex. Some of the outer cortical cells become meristematic. They become meristematic and they become active. They start dividing. They regain the capacity of cell division. They start dividing and they form tissues towards inside of the cock cambium and outside of the cock cambium. See here, this image is very small. You can see. So the outside, this is still. This is still. Let me increase its size. Let me increase this size. See, now you can see it is now clear. This is because of the intracellular secondary growth still is increased. Still is increased. A secondary xylem and secondary phloem are formed. And epidermis is ruptured. This broken epidermis you can see in the image. This is ruptured epidermis. And the outer cortical cells become meristematic, forming cock cambium. Like vascular cambium, cock cambium also produces daughter cells towards the inside and towards the outside. The daughter cells, the cock cambium is also called as, the other name is phellogen. I'll repeat once again. Cock cambium is also called as phellogen. Phellogen. Cock cambium is also called as phellogen. Phellogen produces new cells towards inside the cock cambium ring and outside the cock cambium ring. The cells, new cells which are formed from cock cambium towards or phellogen towards inside become secondary cortical cells or secondary cortex. That is also termed as phelloderm. Phelloderm. The daughter cells which are formed outside the phellogen, that is cock cambium, become cock cells. Cock cells are also called as phellem. Phellem. I'll repeat once again. There are three different terms are there. Phellogen, phelloderm, phellem. Phellogen is a cock cambium. It is formed from the it is formed from the outer cortex. Just below the epidermis, some of the cells of outer cortex become meristematic. They form phellogen. Phellogen is nothing but cock cambium. Cock cambium forms daughter cells towards the inner side, towards the middle cortex, and towards the outside, towards the epidermis. The daughter cells which are formed towards the epidermis become cock tissue. And the tissue which is formed towards the inside, towards the middle cortex, become secondary cortex. The other name of secondary cortex is phelloderm. Cock cambium is also called as phellogen. And the cock tissue which is formed outside towards the cambial ring is called phellem. From outer to inside, phellem, this is the sequence of the arrangement of tissues: phellem, phellogen, phelloderm. I'll repeat once again. From outer to inner side, phellem is the outermost tissue. It is a cock tissue. And the meristematic tissue is called phellogen. Inside the phellogen, there is a secondary cortex. That secondary cortex, the other name of secondary cortex is phelloderm. Together, this phelloderm, phellogen, phellem, they lie outside the middle cortex. They act like protecting layers of the secondary stems. They act like protecting layers of the secondary stems. So this is about extrastellar secondary growth. Outside the stele, there is a secondary growth. Because of that, the new protecting layers are formed. Okay. When the epidermis is ruptured, along with the epidermis, The cuticle layer, which acts like a protecting layer, and stomata may be there in the primary dicot stems. Those stomata are also damaged. Stomata are also damaged. In place of stomata, in secondary stems, some other aerating pores are formed. New aerating pores are formed. Let us see them. Let us see them. So this is the secondary stems are also have aerating pores. Those aerating pores are called lenticels. So this is the enlarged portion of cock tissue. In the cock, you can find aerating pore. This aerating pore is called a lenticel. Lenticel helps in exchanges of gases in secondary stems. These are the empty air spaces are seen here. Air diffuses into the stem through these pores. Because in primary stem, stomata help in exchange of gases. Those stomata are ruptured along with epidermis. Then, due to increased stellar growth, stellar size, epidermis is ruptured. Along with the epidermis, stomata are also damaged. In place of epidermis, new protecting layer is formed. That is phellem, phellogen, and phelloderm. This tissue is called periderm. Together, I'll repeat once again. There is a new terminology. Don't get confused. Phelloderm, phellogen, phellem, together known as phelloderm. Phelloderm is a protecting layer of secondary stems. Outside the cock cambium, there is a cock tissue that is called phellem. Cock cambium is active, which is forming continuously daughter cells, new daughter cells towards inside and outside. The outside daughter cells become cock tissue. The inside, the tissue which is formed from cock cambium, that is phellogen, is become secondary cortex. The secondary cortex is also called as phelloderm. Phelloderm, phellogen, phellem, together known as periderm. Periderm. Periderm is a new protecting layer in secondary stems. In periderm, you can find these aerating pores. In periderm, you can find the aerating pores. These aerating pores are called lenticels. Lenticels lenticels help in exchanges of gases in secondary stems. The loosely packed tissue is seen here. That loosely packed tissue is called simple parenchymatous tissue. There's one new name. There's loosely packed tissue is there. That is epithem tissue or something. There is parenchymatous tissue. Simple parenchymatous tissue is seen here. So this is about secondary growth in dicot stems. Let me sum up. See here in detailed structure, you can see this. So this is secondary growth. So in 2 years, such type of issues are seen here. Let me check. See, this is a strip of cambium is seen here. This yellow color strip is cambium, vascular cambium. Inside there are two woods are there here. Two woods. Because this is 2-year-old stem. When you observe the 2-year-old stem, you can find two annual rings. Two autumn woods, two spring woods. So this blue color strip is there. That is secondary ray. This is xylem ray. This is phloem ray. This is phloem ray. This is xylem ray. Xylem ray and phloem ray, together known as secondary medullary ray. Secondary medullary ray. Let me sum up. Secondary growth in dicot stem. Secondary growth in dicot stem is categorized into two categories. The first category is intrastellar secondary growth. The growth inside the steel is called intrastellar secondary growth. Intrastellar secondary growth is by vascular cambium. Vascular cambium is formed. Vascular cambium is formed by the fusion of fascicular cambium and interfascicular cambium. Fascicular cambium and interfascicular cambium. As a result, a ring of cambium is formed inside the vascular bundles. That is called vascular cambium, ring of vascular cambium. This ring of vascular cambium produces new daughter cells towards inside and outside. The more number of cells are formed towards inside and less number of cells are formed outside. There are two different types of initials will be there. Fusiform initials, the daughter cells which are formed from fusiform initials develop into secondary xylem and secondary phloem. The daughter cells which are formed from ray initials become xylem ray and phloem ray. So simple. Together, the xylem ray and phloem ray are called secondary medullary rays. They help in radial transportation. Secondary medullary rays help in radial transportation. The more amount of xylem is formed, less amount of phloem is formed. That means the secondary xylem is more, secondary phloem is less. Secondary phloem. And because of the formation of secondary vascular tissues, the primary vascular tissues are ruptured or replaced by secondary vascular tissues. And the new tissues are added to the steel. That's why the size of the steel increases. The girthness, the radius of the steel increases. When the central column size increases in the stem, it pressurizes the epidermis. As a result, epidermis is a single layered structure which protects the primary stem that will be ruptured because of the intrastellar secondary growth. Intrastellar secondary growth. Because of intrastellar secondary growth, the size of the steel increases. The increased size of the steel ruptures epidermis. When the epidermis is damaged, in place of those epidermis, that particular epidermis, new secondary layers, protecting layers are formed in place of epidermis. Even along with the epidermis, some of the cortical cells may be also damaged. And the cells which are exposed, the cortical cells which are exposed become meristematic in extracellular secondary growth to produce new protecting layers. In extracellular secondary growth, cork cambium is formed. The purely cork cambium is secondary meristem. But vascular cambium, so part of vascular cambium is primary meristem, having primary meristem. And the part that is fascicular cambium is primary meristem. Interfascicular cambium is secondary meristem. Both primary and secondary meristem strips together combine with each other, join with each other, forming a ring. But in extracellular secondary growth, the entire cork cambium is a secondary meristem. It develops from secondary cortex. It develops from secondary cortex, cortical cells which are exposed. That becomes cork cambium or phellogen. The phellogen produces new cells inside and new cells outside. The cells which are formed outside called phellem, inside phellodome. The other name of phellem is cork. The other name of phellodome is secondary cortex. Secondary cortex, cork cambium, cork tissue, together known as periderm. Periderm is a protecting layer of secondary stems in dicot plants, even in gymnosperms. In periderm, you can see here and there like stomata, aerating pores will be also there. Those aerating pores are called lenticels. Lenticels are wide openings. They don't have any God cells like stomata. In stomata, you can find God cells. That some loosely packed cells will be there. That's it. They help in exchanges of gases. So with this, annual rings you need to remember. That is important for bit. Annual rings are the rings. Uh, they are by counting the annual rings in the secondary stems, we can estimate the age of the plants. There is a special branch in botany is there that is called dendrochronology that deals with the age of the trees, large trees, old trees, by counting the number of annual rings. Annual ring is a growth ring. The other name of annual ring is called growth ring. Both spring wood and autumn wood constitute annual ring. That both the rings represent the wood which is formed in a year. In a year. Wood is nothing but the secondary xylem is also called as wood. Secondary xylem is also called as wood. So with this, let us conclude here. Let me stop. So all the best. So let me close the session.